and we're at the International Mycological Congress number 12 in Maastricht, Netherlands, and it's the 13th of August 2024. And I'm going to talk to someone called Conrad Schoch, who is a very good mycologist, but he's also very good at data. So he's going to tell us about his life, where he was born, how he moved around a lot, and what he's doing now. It's a very, very interesting job in mycology, and one that's helped all the mycologists very much. So Conrad, introduce yourself.
I'm Conrad Schoch. I'm currently the team lead for NC Biotaxonomy, so I'm the team lead of a group of 8 taxonomists that deal with all the names that come into GenBank and other public databases. It includes the European database, EBI, and the Japanese database, DDBJ. So that's my current position. Previously, I was a mycologist, so I think I'm more of a librarian now than a mycologist, because I'm rearranging other people's data, but it's still something I enjoy.
That's very helpful.
Yes.
Very helpful.
Yes. So what else do you want me to continue on? What else do you want to know?
Well, you were born in South Africa?
Yes, I was born in Stellenbosch, South Africa. My dad was a geologist at Stellenbosch University, and then when I was finished with primary school, we moved to Bloemfontein, which is in the center of the country, the Free State. And I started my studies at the Free State in the Department of Microbiology. At the time, Mike Wingfield was there as well, but I was not a student of his. But his wife Brenda was there too. So Brenda Wingfield, I think, made a big impact on my life without being my advisor. I had kind of a tricky master degree that I had trouble completing, which I did complete. I was working on Saccharomyces cerevisiae, stress response in Saccharomyces cerevisiae. And she actually, called me aside and said, there's this guy in Stellenbosch University called Pedro Kraus. He's looking for a PhD student. Are you not interested? And at the time, this seemed like a really good opportunity. So I packed my bags after completing my work on my master's, and off I went to Stellenbosch and started a PhD with Pedro Kraus, which was a really good choice in the end, because I also met my wife in the lab there. And from there, things went very well. I enjoyed my PhD.
What'd you work on?
I worked on Calonectria. So Pedro actually did a lot of sampling. He would go off to Brazil, and then he shows up with these bags of soil. And then I baited the Calonectria species out of it. So I did some mating studies. So we could find 4 different species, which I described at the time. And also several other things that were in the lab, that we isolated Calonectria from. So that was essentially my PhD. And after that, I got a postdoc at Cornell University with Jim Aist. But there was also Gillian Turgeon was also in the lab, and they went to San Diego. It was Novartis, I think, whatever the company was before Novartis, started a research institute. And they invited me to come and continue my postdoc there, because I think she was partially responsible for the grants. So I was in Cornell for 3-4 months in the middle of winter. I went back to South Africa, got my PhD, got married, came back with my new wife, Barbara Roberts, who's also a mycologist, actually a plant pathologist. Went to New York, upstate New York, and then immediately to San Diego. And I did my postdoc there at a company for, I don't know how long, 3 years we were there. I'll have to check, but It was an experience. I mean, we were it was basically the first genomes that were sequenced there. So you would go into the lab. So it was a commercial lab. I'm used to academic labs. The first office you see was the patent lawyer they were sequencing the rice genome. So they were patenting all the genes in the rice genome. At the same time, they also sequenced several bacterial genomes, as part of the rice genome, that just by happenstance. And then also some of the first fungal genomes were sequenced there. So I was not directly involved. I was still working on my Cornell project, which was trying to transform Nectria, Metacocca, Fusarium solani, looking at motor proteins, making mutants and describing motor proteins. There was a company producing an enzyme that was used for transforming that species, Novozyme, and they stopped production. So essentially, in order to complete my experiments, I needed to figure out a way how to transform that species, which I never really did. So my postdoc was kind of unsuccessful, but I got to play around with lots of genome data at the time, totally unexpectedly. In the end, I actually produced a literature review on motor proteins from genome data without actually doing a lot of experimental data. So I think I was lucky in a lot of places, being at the right place at the right time without really planning it. So to continue from there, so Novartis got merged. No, whatever was before Novartis, I can't remember.
I don't
the companies.
remember either.
Yeah, it became Novartis.
Yeah.
They merged, and that whole research institute was disbanded. So that was a really interesting time because people were trying to get their resumes ready, and there was a lot of really good scientists there. One of the interesting things, so they had a lot of money, right? So they basically built a robot to do single spores for fungi. The robot never worked. In the end, we ended up hiring an intern to do it. But the engineers spent a year building the robot. I think they got the robot to make cocktails. It could pour cocktails, but single sporing. It was also a cell sorter. We bought a cell sorter to do the single sporing. The spores were too big for the cell sorter, so there was some. But it was an interesting time. Others should talk about it more. I was sort of just peripheral to that, but so that finished very quickly. But suddenly we had a whole network of people, and we also learned a lot. Then the postdoc opened up for AFTL, which was another being at the right time, right place, right time moment.
And AFTL.
So
you know
AFTL was
an acronym for?
Fungal, assembling the fungal tree of life. It was the first grant. Joey Spadafora's lab in Corvallis. So I had a colleague and a friend in Portland that we went to stay with for the interview. So we drove up to Portland, and then the next day I drove down to meet Joey. And so I always remember Joey's son opened the door Saturday morning, and Joey's nowhere to be found. And
Had he forgotten?
No, they had a party the night before.
Oh, he was upstairs in
So
bed.
they were, yes, they were saying goodbye to a student. So anyway, but it was a great lab, I started there. We did a lot of sampling for assembling the tree of life. I could go visit the Netherlands. Pedro, by then, was my previous advisor, was head of CBS. Mm-hmm. So I could come in and sequence some of the cultures from CBS. So that was.
Yeah, CBS was very generous to that
project. It was an
project.
essential part of that whole project, I have to say. If it wasn't for the CBS cultures, I think so the project, everybody had different parts, and my part was non-lichenous Ascomycetes. Mm-hmm. So CBS was essential in that part of the project. I have to say, some of the other parts were done by different people. So it was really a team effort. So again, being part of a team, that's something I learned there. Also, being dependent on other people to get the stuff, to get the material. that was all very important. So, it was a really wonderful project to be a part of. I think it was, sorry.
I was just going to say, yesterday we were having a little bit of a discussion about whether people who do the collecting of the fungi should be on the paper.
Yes.
And that's an important question.
So, I think there was a lot of discussion also when I was at the research institute, the Torrey Mesa Research Institute of the company, about how, there was a lot of fights about authorship and who should be first, who should be second. I learned there that it's always good to get the authorship sorted out really early on and not when the piece of work is done, because it can be really problematic.
And I think with the fungal tree of life, assembling the fungal tree of life, it was important to let people know that if they did contribute specimens, they would be included.
That was a big part of it. So we had long author lists, and we would make sure that people knew that everybody contributed to that. I continued that kind of large collaborative effort in some of the other work I did afterwards, because I found it, it was really rewarding. Being sort of a linking person without being the only person producing the data. So we did some other papers like that. The barcode paper was another one we did, where we had input from a lot of different people. And again, without their data, the barcoding paper would never have. So I can explain about the barcoding paper.
that would be good. You say you're a librarian, and
yes,
so,
I think so.
You're more than that.
Right,
but it was very important for you to be able to bring everybody together and look at all these different DNA regions. And
I think I feel a little bit like Forrest Gump. I think I've been at the right place at the right time, finding doing work that others didn't want to do at the time. Okay, so from Oregon State, so actually, there's a lot of stories I can tell about Oregon State, and Joey had a huge impact in our life, in our careers. We had a grant there with Joey's help. We got our first grant, and that was really important. And then the job opening came at GenBank, so they were looking for a fungal curator, so I got that position just in time because I had a young son and I needed to get a real job. So I think it was Keith Seifert that asked me about the barcoding paper. It was just, I think there was a need for somebody to just push this thing toward a place where it could be published. And a lot of people were really willing to share data. So, again, we had lots of different people coming together. It was right place, right time. People were willing to share stuff. The analysis were done by several people as well. And I think my main job was just to link all the people together and make sure we get the thing done. Keith is, before that, Keith Seifert did a lot of the groundwork, produced some data himself, comparing different loci. And for the barcode paper, which turned out to be, I don't think anybody foresaw how big an impact paper it would be, because it was almost a foregone conclusion that ITS was going to be the locus, because it was already being used by the environmental community. And we just needed to write up something to compare it to some other loci so that actually there's some real data to back it up.
Now, the only problem with that was I was working with yeast, and everybody working with yeast had established the large.
16, yeah. Yes.
So
yes,
anyway,
but it was really
it
was the environmental community that actually wanted the ITS,
and it's worked out. Now there are exceptions, but in general.
Yeah. So we different people also developed some of the protein coding markers like RBB1, RBB2. Some of them are still in use. Elongation factor. Different people provided the primers, So that was all came from AFTOL, and then we used some of the AFTOL markers to compare against ITS. So I think that's probably the paper that got the most impact that I was involved with.
It's got lots of citations.
But it's, again, I'm first author, but it's
well, you were the cat herder, as we say.
Yeah. That's essentially the
but that's
job.
a big job. So what are you doing now? And I just can't imagine how you keep up with all the names.
I don't. I don't. So I think we do the best we can. So, you know, I think, you know, we need to find better ways to discover, to actually use the literature. That's still a big challenge. The best I can do now is to work on the names that I see daily. I mean, people submit names. I see new names coming in, and then I look at a name, and then I discover something about something that's related to the name. You know, so it's sort of like a butterfly going from flower to flower. So there's no real method to the madness. There is some sort of a following of the literature for big picture kind of papers, and coming to a conference like this is important. I can see important papers, people discussing new classifications based on genomes, so that's important. But it's a challenge, and you know, we have to cover, especially the invertebrate guy, has to cover, you know, 2/3 of all names being you know, insects or arthropods. So I can see similar. Being a team lead now, I'm always, you know, interested in fungi, but I can compare fungi against some of the other groups as well. So I think it's important, and this is something that somebody told me, that looking at the bigger picture. So I think you're focused in one thing as a scientist, but scientists also need to take a step back and see how their research fits into the whole ecosystem of other research. And I often find that very accomplished people they're just interested in their neck of the woods or their set of names. They don't care about if it affects names related to it. So I think people need to consider that too, because even if that's not of interest, you're interested in this group, all of these things do have connections. And it's very hard to just carve out your own niche and think it has no impact on anything else.
Well, you certainly have helped everybody, all of us, because I always, you know, if I have any questions about what right name to use for something, I often check GenBank first.
I've just been in a session where NCBI taxonomy came, I think, third or fourth behind Mycology and Index Fungorum as a place where people look for names. We have a disclaimer that says we shouldn't be used as a primary resource. We still prefer that people go to those, because we rely on them as well.
I know.
And I think it's important for us to acknowledge these primary resources, and ensure they get funded and supported. So we wrote a paper in 2020 where we actually listed all the resources that we use.
No, and I do, well, when I was doing it, I'd use other resources, but I would always, it was a shortcut to give me an idea of what to do.
So you know, even having that disclaimer means people still use our names. So the responsibility is still on us to do as good a job as we can.
Well, I think you do a great job.
Well, there's always room for improvement, so we always want to get better.
So one thing I wonder about in that sense, though, is how many people are working with you that work on the fungal database?
Okay, so I'm the only fungal person that's responsible for all of fungi. And then, you know, there's somebody working on viruses, there's somebody working on plants. We just got a new person working on plants. So that's it. I mean, that's.
So it's you by yourself. You don't have any help?
No. No.
Whoa.
No, we do have. I mean, we do have developers. We have 3 developers that builds the database and makes sure the database runs. You know, keeps the maintenance, and you know, database evolves, build the tools and stuff like that. But for content moderation, I have to say, Barbara Roberts, who also works at.
I was going to ask you where Barbara was.
She's also at NCBI. She's working at RefSeq, which is the curated version of GenBank. So it's a separate database. It takes GenBank records and then it curates it, so it can change things on that. Normally not the taxonomy, but it will annotate genomes. So that's. It's actually, a lot of people rely on RefSeq instead of GenBank because it's sort of a separate selected set of records. So she's the fungal genome curator for RefSeq, and she often sees classification issues by analyzing genome data, and then she makes tickets for me. So, you know, I need to fix this or that, or I need to add type material to this because she's seen it. So she does a lot of those incidental things that she sees from the data, because In taxonomy, we only look at the names, we don't look at the sequences as much. So we only care about names, and the names are the central organizing database at GenBank and beyond. Actually, not GenBank. There's the chemistry, PubChem, the chemistry database at NCBI uses it to organize things. Various other databases uses it beyond GenBank. So it's an important database. I think it feel a bit like being the water board. If something breaks in that database, people notice. But if you do your job, they don't. So that's exactly how it should be.
Well, you certainly do a huge service.
Well,
And you are
I'm
a very good mycologist, which makes it usable.
well, yeah. Like I say, I rely on other people's good work to expose it. So I think that's my job now, is to ensure that other people's data get treated appropriately.
Well, it's good to see you.
Yes.
And you need to come to more MSA meetings.
Yes, I will.
Thank you.
I will. Yes.
Thanks.